Good afternoon, everyone. On behalf of IIT Madras, welcome to Healthcare Magnified, a virtual summit for corporate leaders. I'm Padma Thumandram. I handle marketing for Alumni and Corporate Relations at IIT Madras, and I'm glad to be your host for this program. IIT Madras is and has been India's #1 institution in innovation ever since the prestigious ARIIA rankings came out. Over the next hour and a half today, some of the country's best technology experts at IIT Madras will be sharing about their innovations in the healthcare domain with you. Before we proceed with the program, I'd like to warmly invite Mr. Kaviraj Nair, the Chief Executive Officer of the Office of Institutional Advancement at IIT Madras, to formally welcome you all.
Good afternoon, everyone. So I think the agenda is pretty clear, and we will get into specific sessions on healthcare-related areas. But before that, I just want to take this opportunity to welcome all of you to this session, and also give a 2-minute overview about IIT Madras, some of the key areas that we work on at an organizational level, before we kind of proceed further with specific sessions aimed at with a specific agenda and areas. So as Padma mentioned, IIT Madras has been at the forefront of technology-driven social transformation in the last few years. We, in the last several years, we have developed centers of excellence, which are interdisciplinary in nature, and which kind of address specific social issues, and look at holistic solutions in that level. So if you look at some of the, we have also been more research oriented in the last few years. Our PhD intake has significantly increased. Previously, we were as an institute, we used to give more priority to the undergraduate programs in the past, and that has completely changed in the last few years. The focus has been clearly on research areas, and we have about across the 4 or 5 years, we should be having about 2,800 PhD students, across the streams, which itself is shows the shift in focus that IIT Madras has been having in terms of more research-oriented programs or more patents and things like that. So we have got more than 1,000 patents in the last few years, and the research spans across multidisciplinary areas like healthcare, water, sanitation, environmental technologies, and all of that. Of this, healthcare certainly occupies a supreme position, both at the highest end of research, which include things like molecular research, molecular medicine, or like in the cancer research or things like that, and at is socially mass-specific innovations, which can address larger problems of the society, like it could be in terms of contactless, diagnosis or low-cost diagnostics, which will have mass impact and which could transform the whole rural economy in many ways. And we do have sessions around that also, right? And I think that is pretty much what I wanted to give as an introduction. Thank you again for joining the session. And before I hand it over back to Padma for specific sessions, because I don't want to take much of time here talking at the overall level. I'm sure that you're all waiting for individual sessions. So let's proceed with that. Happy to kind of work with each of you. I think the idea is to clearly showcase some of the technologies that we have which are highly innovative in nature, which can have a transformative impact on the society, and also to see what are the ways in which we could work with you. And as you would as I'm sure that most of you are aware of, clearly the CSR funds can be used for scientific research. The statute allows us that, like, if you, the Minister of MOCA actually, Minister of Corporate Affairs actually allows that, the CSR funds could be used for scientific research. So clearly that's an area we have done in the last few years. We have created partnerships, very successful partnership with clear milestones and deliverables in this area of scientific research. And our teams will work with you to kind of, understand your scope out the specific problems that you would want to work on. We'll scope out milestones, deliverables, all of that. And being an institution of this stature and eminence, we have a very clear and transparent way of governance structure that we put in place, tracking mechanisms and milestones. And we're also very keen to understand how this is impacting the whole thing. So we also do audits to figure out what are the number of lives it has impacted, what is the overall impact on the society. So clearly driven by metrics. I think these are some of the points I want to mention. One is obviously the content of the work, and beyond that, we also measure the impact very clearly with clear governance structure. So that makes it even more easier for you to understand that the overall impact you have created. I will not take any more time. I will hand it back to Padma. Over to you, and thank you everyone again for your time. Hope you have a very fruitful session going forward. Thank you.
Thank you for that. So I was just thanking Mr. Kaviraj for the warm welcome and for setting today's program in a comprehensive context for all who have logged in. I now have a small request to share with the corporate leaders who have joined in for today's program. Could you please type in your name and your organization's name in the chat box, so we would be able to, personally welcome you as well to the program, right? So in terms of format for the program, we have 3 panels that are lined up for you. And all of these 3 panels feature some of our key work across healthcare that we felt would be of interest to you. The first panel is on oncology, the second is on drugs and formulations, and the third panel will be on healthcare equipment and infrastructure. Now, each of these panels would be for a total of 20 minutes. It will be followed by a time for questions from the audience for 10 minutes. So in case you have any questions, feel free to type them into the chat box as the session is going on. And we will do our best to address all of the questions at the end of each of the panels. Otherwise, we will get back to you personally post the event with responses to your questions. So without any further delay, I invite the team from the first panel, Oncology, led by Professor Mahalingam, to come forward. So over to the opening batsman of panel 1, Professor Rayala Suresh Kumar. Over to you, Professor.
Okay. Good afternoon, everybody. So I'm going to just give you a brief background on what we do and what are our research interests. So our interest is developing small molecules, peptides targeting novel oncogenes. And we established the molecular mechanism of action of new drugs, use the recent technology CRISPR-Cas9 screen methodology. And we also look at studying mechanism of therapeutic resistance. And finally, our core crux of the work is developing preclinical models of tumor progression, which includes xenografts, patient-derived explants, and patient-derived xenograft models. So this is the core research work that we do. And in addition to that, we also do some translational work. So we have good expertise in generating monoclonal and polyclonal antibodies. And these antibodies, we use it for making diagnostic kits and diagnostic applications. So move on to the next slide. So this is what is the establishing PDX and PD model. These models we use to look for drug screening, and this will be used for developing personalized therapeutics. We just started developing these models, and this is from understanding the molecular pathogens of cancer, and for developing personalized therapeutics, we use tissue from the patient, direct tissue, of course, of ethical clearance, and then we inject it to the mice. And then we develop a series of mice, like first transplant, second transplant, third transplant. And then we divide them into groups and treat them with the drugs. And then we look at the drug response of the animal. This is with the patient-derived explant models. Whereas in vitro also, we patient-derived in vivo. Whereas with the in vitro, we culture the cells and then we treat them with drugs in vitro situation and harvest the tissue and go for a marker analysis. And finally, we look at the different markers. So these are the 2 different models that we have established, and it's ongoing. And coming to the work that we translational work that we have done. Next slide. So there are 2 translational research projects that we have from our lab. The first one is the cervical cancer detection, which is a sort of a point of care device. And this we tested it in for HPV 16 and 18 detection in rural women. And this is a sort of bench to bedside application. We have developed a self-sampling kit, and this self-sampling kit will be given to the women where in rural areas where they don't have access to much access to the hospitals. And then they collect the sample and then give it to us. We do a LAMP PCR, and with the help of this handheld fluorescent detector, we could see within 30 to 45 minutes, we can tell if it is HPV positive or negative based on the paper-based method. So this is sort of point of care device that we have developed, and we are testing it now. Next slide. So this is a paper-based, the same thing that I was trying to say, paper-based lab-on-chip device for detection of high-risk HPV. So our objective is to identify the HPV infection cervical fluid samples using LAMP PCR. And second objective, to develop paper-based material and fluorescent dye for HPV PCR amplicon detection. And third one, we fabricate fluorescent-based device for detecting the signal from paper-based system. So this is how it looks like. So with this, we achieved these three objectives, and now we have the device in place, and we are going to test it out on the rural population. We have a patient target of 10,000 patient, and then the same samples we'll be using for pap smear test to detect if there's any correlation between these two, between this device versus the pap smear test. So our outcome, whatever till now we have done, 129 samples were screened and PCR amplicons were tested. And paper-based detection system and dye was confirmed using spectrophotometer and tested in device. And device can differentiate and detect HR positive versus HR negative. This is the device that we have done. It has an eyepiece and 2 filters, and then it has a white LED light source. Next slide. So how it is important, or what does it add to it? This probably this test takes approximately 30 to 45 minutes, whereas if you have to go with the conventional pap smear test, it takes close to 1 hour, at least the 1 or 2 days, and it requires a pathologist to look at the slide and then give the report. Whereas here, just based on the color change or based on the detection method or color change, a technician, simple trained technician can tell if it is positive or negative. So we have this device in place, and we are ready for the trials or the field trials. Oh, thank you so much. And if there are any questions, I'll be able to take it up.
So any questions, we will take it up all together right at the end, Professor. So let's maybe go through
now, probably I'll request Professor Mahalingam from National Cancer Tissue Bank to present his one.
Okay, good evening to all. I'm Mahalingam. I work in the Department of Biotechnology and National Cancer Tissue Biobank at IIT Madras. Our lab is mainly concentrating on basic mechanism on tumorigenesis as well as and cancer genomics. And the basic mechanism, basically we work on like a GTP binding protein. That is, we were the first one who identified this group of protein. It's present in the nucleus and it's involved in maintaining stem nature of the stem cells. Now we are looking at the whole mechanism, how it maintaining the stem nature of the stem cells and how we can use this as a like a diagnostic purposes. And also we are working on the RAS protein, try to understand, because we all know that it is an untruckable oncogene, and it's most of the cancer, you have more than 60% of mutation in this tumor, and everybody's looking for the inhibitor for this. And we are trying to understand the whole mechanism. And right now we have identified a couple of small molecule inhibitors as well as peptide-based inhibitors. And we checked in the cell lines. It is, I mean, we can able to see very nice inhibition of the tumor. But right now we are moving to animal models. And next one, please. Next slide. So the other interest is the previous slide. The other interest is the cancer genomics. Here, we all know very well that in India, we don't have much information on genomics of cancer patients from Indian population. And then whatever the treatment we are giving right now and whatever the diagnostic tool is available for the treatment and management of tumor, everything is developed against the Caucasian population or the Western population. That is why we don't see much success rate like what we see to the Western population, because we need to know the genomic intricacies of our own population, and based on that we have to develop the marker and identify the targets and go on for the novel therapeutics. Keeping in that mind, we have developed or we have established the National Cancer Tissue Biobank. It's one of its kind in India. It's a community-based biobank. So in this biobank, so far we have collected more than 5,000 patient samples from different cancer, collaborating with the different hospitals in Chennai and Pondicherry, including JIPMER. And using the tissue samples, of course, all the tissue samples collected after ethical clearance and informed consent from the patient, particular patients. And we have initiated the comprehensive genomics of the cancer which are prevalent in India, like for example, breast cancer and oral cancer, and we started with the pancreatic cancer. We have sequenced more than 200 patients, both control as well as tumor tissue, as well as the germline DNA from PBMCs of the same patient. And the preliminary analysis is very encouraging, and we can able to see the uniqueness of the Indian population. So for using this information, we are planning to develop the biomarker specific to our Indian population. Next one, please. So in addition to the tissue samples, we also collect the blood samples as well as the urine from the urogenital cancer patients. And this blood sample, particularly, we concentrate on cfDNA, because you all know that it's a free-floating DNA in the blood. And if you can able to correlate whatever the changes, whatever the genomic alteration we identified from the tissue samples, if you can able to see it or identify it here, that can help you in multiple ways. Like, for example, you can develop a non-invasive biomarkers, as well as we can able to classify different tumors just based on the cfDNA. It's floating around in the blood. So next one, please. And in the untrackable oncogenes, as I said in the beginning, that we mainly focus on the RAS and MYC, because these two genes are required for even initiating the tumor, because the transformation and immortalization, these two are involved. Plus these, basically these two genes are required for the normal physiology of the cells. That is why we are not able to target these to prevent or to inhibit the tumor formation or tumor progression. So since these two are very critical, and also if you look at the picture very carefully, there is a crosstalk between these two. And using this strategy, we have identified a peptide particularly for a MYC protein. And we have tested in the cell culture system. We can able to see very nice inhibition of the cell growth. Now we are going to the animal model. Next one. So this is basically the normal physiology, the physiological role of the MYC, because in the tumor, you see multiple kind of abrogation. You see amplification as well as a mutation. So particularly if you look at the neuroblastoma, you see like a very high level of amplification. That is why first we targeted MYC rather than the RAS oncogene. Next one. So if you look at very carefully what the kind of treatment are going on in India or the most of the places, so it's like kind of one size fit all kind of therapy. So without knowing or without having much information on the genomics, we just give whatever the drug is available. That is why you see different kind of effect in the patient. So you have adverse event, you have no benefit, sometimes you get the benefit. That's why the success rate is very low. So whatever the information we collect or we generate from the basic science as well as the genomics, what we are doing, we can able to stratify the patients towards the personalized medicines. So this based on the marker, whatever you identify, based on whatever the target you identify, and you can target the population towards the precision medicine, which will give you much better survival rate as well as the disease-free survival. That is what is going on. If there is any question, please feel free to ask me. Thank you.
Thank you, Professor Mahalingam. The audience, you're free to post your questions in the panel, in the chat box. We will now have Professor Nadia Muttaragu from the Biotech Department to share her presentation with us.
Yeah.
Over to you,
ma'am. Hello, everybody. I am Nadia from Biotech Department. Just to give you a brief background about my expertise. So I did my PhD in University of Glasgow, UK, and stayed there for my postdoctoral training. I did my postdoc in Beatson Institute, Cancer Research UK Institute. Both during my PhD and postdoc, I have extensively used cancer models, both in vitro and particularly transgenic mouse models, to answer both basic and translational question. For example, I have developed and characterized new mouse model for pancreatic cancer, which is driven by cancer promoting genes like MYC and KRAS. And what we found is in this model, both these oncogenes drive tumor progression by escaping immune system, particularly B cell and NK cell mediated immunity. On different project, I used different set of mouse model to drive colon cancer. And what we validated is one of the kinase in the AMPK pathway called NOAC1, which is specifically able to kill cancer cells, particularly MYC overexpressing cells compared to normal cells. So both these studies were published in high impact reputed journals. And one of the things that was key in all these studies is we were able to modulate our favorite oncogene or tumor suppressor in the particular tissue that we are interested in. Next slide, please. With this background, I established my lab just over a year ago, where my lab focuses on three main areas. First, we want to understand mechanism of cancer progression. We know that cancer metastasis and resistance to available chemotherapy is one of the major roadblock in improving patient survival. So what we are focusing on is to understand plasticity of cancer cell, which is in a simpler words, ability of cancer cell to change from one cell type to another cell type, and their contribution to metastasis and chemoresistance. So we are looking at ways of targeting this plasticity, thereby improving with the aim of improving clinical outcome. Secondly, we are also running several synthetic lethal screen to identify therapeutic targets for pancreatic cancer to begin with, but we will follow this up with other cancer type also. What do I mean by synthetic lethal screen? If you know the current chemotherapy spectrum, the major roadblock is the side effects because of the toxicity these drugs have on normal cells. So with this synthetic lethal approach, what we are aiming to identify a nodal point, which is specifically kind of important for cancer cells, but not important for normal cells. So we can identify drug target to specifically target cancer cells. The third important aspect that I will brief upon today is to establish preclinical model for various cancer. Again, next slide, please. The ability to kind of identify drug target or improve the patient outcome depends on availability of a preclinical model that represents human patients, that also represents diversity, tumor heterogeneity that we see with human cancer patients. So the idea here is to get the tumor patient from the sample, dissociate into single cells, and then culture them in a 3D environment as an organoid. So because of the ease of establishment and the short time frame it requires to establish this tumor culture, we can sequence this, either sequence this organoid, or as Professor Mahalingam mentioned, sequence the initial tumor patient to identify mutation, and then we can test the drugs, whatever drug based on our mutational burden, and then kind of this feedback can be, this can be feedback into the clinical setting. So since, as he said, that we have a one-size-fits-all approach, this kind of developing individual model for individual patients will drive precision medicine or more move towards a personalized therapy. Yeah, thank you. Yeah, if you have any questions, we are happy to answer.
Thank you very much to the oncology team. Now, any questions from the audience? We can take it now.
Yeah, I think there is one from.
Yes, there is one question.
Yeah, so have you tried to understand the relevance translation of PDX and PDX to human? Yeah, we just started doing this, we just worked out for the clinical samples. So we are yet to test them and look at the relevance to the treatment that's ongoing or that's going on. And also on cervical tumor topic, have you compared your screening test with pap smear? Yeah, for a few samples we have done it and it's matching with that, but our goal is to test at least 10,000 samples, and then the same samples will be given blindfolded to pathologists for pap smear. So from that, we are doing it now. And from that 10,000 samples, if we compare with the pap smear, we'll get to know how accurate is the test. But definitely, we are going on a positive note that so far we have tested 120 samples, and out of which I would say close to 114, 115 samples have come up positive in both the screens. So I think we are going in the right direction with the kit, with the handheld device that we have.
I think next question is for Professor Mahalingam.
Professor Mahalingam, are you there?
Yes.
Okay.
Just saw that.
Sure.
Yeah, the question is, precision medicine is a great initiative. Why not increase the biobanking to other states also? Yes, initially we started with the Chennai, and now we started collecting samples from Pondicherry, and also that we are in discussion with the Kerala and Mizoram. Yes, we are moving slowly one place to the other states.
Okay.
Are there any more questions? Any more questions? No? All right.
Okay, thank you.
Thank you.
Thank you all. Yeah.
Thank you all. Thank you to everybody.
Thank you, professors.
Thank you.
We will now move on to the second panel on drugs and medications. The first professor to share his presentation with us is Professor Sanjeev Senapati. Professor, you could just see if you're able to, if your mic is working.
Yes, can you hear me, Padma?
Yes, I can hear you, Professor.
Great, yeah.
Over to you, Professor.
So please note that I'm joining through Basa's account. I'm Sanjeev Senapati, but somehow my Zoom link doesn't work. So I lead the biophysics group here at IIT Madras. And two major areas that we work on is to understand the mechanism of action of disease-causing proteins. And after understanding the mechanism, then we go for drug design. And basically, we combine molecular modeling, molecular dynamic simulations, and then validate them by in vitro testing. The other major area of what we work on is the discovery and applications of several green solvents. Ionic liquids is one such green solvent, which I'll be talking today. Also deep eutectic solvent. And here also, here we basically synthesize lot many ionic liquids, we characterize them, and we do in vitro assays and computer simulation techniques. Next slide, please. So what are ionic liquids? So ionic liquids are basically salts that are composed of organic cations and organic or inorganic anions. So, and you have lot many possibilities of combining different type of cations with different anions. And in principle, you can generate lot many such combination. So in our laboratory, we first do computer modeling to generate several ionic liquids. And after we find some particular IEL which has specific application for a specific case, then we synthesize them. So right now in our laboratory, we have a big library of biocompatible ionic liquids that we have synthesized and also characterized by several chemistry, NMR, mass, and different other techniques. So these IELs do have different application in different areas. Next slide, please. And in this talk, I'll be talking about two separate applications. So the first application is how we are using ionic liquid to improve the drug bioavailability. As you know that majority of the drugs, they suffer from low water solubility, they have suboptimal clinical efficacy, and many a times their delivery mode is also not very convenient. And if you look at BCS classification system, biopharmaceutical classification system, so it has been divided into 4 classes. Look at class 4. All the molecules here, all the drugs, they do have low solubility, low permeability. Even class 2 have low solubility. So solubility and permeability is a big question. And to take that, in this work, we have taken 4 model drug molecules. We taken aspirin, paracetamol, and we taken ibuprofen and diclofenac. So what we have done, basically, we took these APIs, which is active pharmaceutical ingredients. We took these APIs and then make the derivative using our ILs. So I'll be talking them as API ILs. So these are the different derivatives we have with IL1. I'm not disclosing what that IL1 is, what is that IL is. So with that IL, we have made derivatives of paracetamol, ibuprofen, diclofenac, and ibuprofen and paracetamol. Likewise, with the other IL also, we made 3 different derivatives. Next slide, please. So after making these molecules, the API ILs, we now checked their efficacy. We checked their bioavailability. So we have looked at their stability at 37 degrees Celsius, 45 degrees Celsius. The parent ASP, parent API is at the beginning, and then all the derivatives are shown here. So you can see at 37, 45, our API ILs are better stable. We also treated, we also had acidic and basic treatment, and we found that our API ILs are acidic and basic stress proof. And most importantly, look at the solubility. So these two are basically ibuprofen and diclofenac. They are BIC2 drugs. And in our API ILs, we have solubility increased by 10 to the power 3 times. So these drugs, which were very poorly soluble, with our derivative, they become 10 to the power 3 times more soluble. Also, their uptake is quicker. If you look at the dissolution, look at this diclofenac. It takes about 60 minutes to dissolve into the solution. But our API ILs, it just goes in no time. For all 3 other drugs also, the dissolution is much quicker. So that means the uptake will be quicker. Next slide, please. But the membrane permeability is not compromised. You can see the paracetamol, parent paracetamol, and this is these are our derivatives. So permeability remains unchanged for all the derivatives we tested. And very interestingly, our derivatives can be delivered in liquid as well as in colloidal formulation. Look at this table. So amount of drug that is soluble in 10 ml of aspirin is 57.45 mg, whereas our derivative with IL is 3747 mg, which is 3.7 gram in 10 ml. So there is a 65-fold increase in solubility, and in these cases, ibuprofen derivatives, it is 1,000 times. So this is in drop form. We also checked in the colloidal form. In colloidal form also, we have 86 to 1,000-fold uptake. So which means is that, instead of 650 mg paracetamol, now I just take 1 drop of paracetamol liquid. And this is very important for children as well as for elderly, where, taking the solid tablet is difficult sometimes. So, with our IL derivatives, the solubility has gone many-fold high, and next slide, please. Formulation also has become easy. So, these 4 were just basically our model drugs. So now we are taking up similar reformulation strategy to improvise the life-saving drugs like paclitaxel, ritonavir. We are also checking curcumin, whether it can be made better, soluble, and can be utilized as drug in future. Next slide, please. So this is one application. The other application is stabilizing DNA, RNA at room temperature using our ionic liquids. So here, if you look at the gel data, so we have stored our DNA samples. This is cup-thymus DNA at room temperature for 36 months and longer. And when we see the control in buffer, so DNA is completely gone, but in our different ILs, my DNA is completely intact. Look at the CD spectra. The DNA in water, which was kept for 36 months, is completely gone, whereas my DNA in IL is very much like DNA in fresh water. And of course, to come up to this experimental data, we had done a lot of computer simulation techniques, molecular modeling, and then from there, we basically have shortlisted which IL molecules to be synthesized and which to be taken up for the experiments. And I'm not showing all those data here. But essentially, this is a combination of, next slide, please, a combination of molecular modeling and experimental techniques. So one can argue that the DNA, what you are storing at room temperature, whether it is functional. We also have done the functional studies taking plasmid DNA. So here we took PBAD, and this is the transfection. We also have transformation studies, and we have seen that some ILs, for example, choline acetate, is better than even the buffer. However, some ILs like BMIM acetate, you see there is no transfection. And why so? Why it is so? We checked the toxicity, and we found that BMIM acetate is actually toxic in the HeLa cell line. Because it's an imidazolium cationic group and having a butyl chain, so therefore it is not very biofriendly, whereas choline is very good. So that is, next slide, please. So that is why you have to understand what application you are interested in, and then depending on the application, we have a library of ionic liquids, and we can utilize the IL for the particular applications. So right now we are basically working on artificial intelligence, so that by AI we can beforehand take up which IL to utilize for my application 1 versus application 2. So similar strategy is underway to stabilize RNA, and also we are looking at several vaccine candidates, if we can stabilize them at room temperature. With that, I stop. Thank you very much for your kind attention. So I'm Sanjeev Sanapati once again, and that's my email ID.
Thank you very much, Professor Sanjeev. Now over to Professor Gopalakrishna, please, from Biotech.
Thank you, and good afternoon to everybody. I hope you can hear me.
Yes, we can hear you, Professor.
Great. So we've been working on calcium binding proteins and G-protein coupled receptors for quite some time now, almost 18, 19 years. Next slide will actually show you the G-protein coupled receptor that we work with. Can I go to the next slide? Hello?
Yes, Professor.
Yeah,
the slide has changed. So, yes.
Yeah. One second. Now let it be there. Hello?
Professor, we can hear you.
Let it be in the previous slide.
Previous. Okay.
Yeah, so we work with what is called as an APJ receptor. One second. What we call as an APJ receptor, because it detects apelin, a 13 residue peptide that is shown at the bottom of the screen. Now, G-protein coupled receptors are very interesting because they're transmembrane proteins, which means they're on the boundaries of the cells, which means part of them, the part that is shown on the upper right side, is outside the cell, and the lower part of them is the inside of the cell. So they're helping cells in detecting signals that are coming from outside and helping cells respond to the signal that the cell has received. So it does a multifarious, these receptors do multifarious functions for the cell, from detecting light, as we see in vision, from detecting small molecules as in taste or in smell, and enabling several physiological functions. Our heart, for example, beats because of a G-protein coupled receptor signaling happening, because it detects adrenaline from outside the cell and eventually causing the cell to pulsate. So the muscle movement that is happening in the heart is because of a signal detection by a G protein coupled receptor. So our G protein coupled receptor, which is the APJ receptor, is involved in angiogenesis, which is blood vessel formation, in cardiovascular development. It's also found a lot of it expressed in the heart and in the brain. It helps in fluid homeostasis, in neuronal cell proliferation, and among other things, it also helps in HIV pathogenesis. And these receptors, while they receive signals from outside, like a photon or a small molecular ion, bivalent ion, calcium, for example, or peptides, as ours does, they transmit the signal into the cell by activating what are called as G proteins. These are called G proteins because they bind guanine nucleotides. So activation of the G protein means it will release GDP and take up GTP. So that's an active state of the molecule that will eventually convey the signal into the cell downstream of the receptor. So what the receptor is doing is it's remaining in off state all the time. And upon receiving a signal, binding of the ligand from outside, it is getting activated itself, which will lead to activation of different kinds of molecules like the G protein and the beta-arrestin receptor. I'll tell you in a minute what that difference is. So here, what is happening is this receptor is receiving signals that are peptides. So it can recognize a 13 residue peptide, or a 17 or a 36 residue peptide, and a 55 residue peptide or a 70. The peptide itself in the cells is made at a 77 mer, but gets chopped into the 13 mer. So the 13 mer is actually as active as the 77 mer. So there has to be a functional relevance as to how come the 13 mer is being made, why the 77 mer is being chopped off, and how we can modify the 13 mer into a smaller molecule to be able to generate a drug. Okay, that's the most important thing. The drug candidate that we should be able to generate by modifying the 13 mer, making it smaller or modifying it chemically, which should help us in understanding the pathogenesis, that problems happening in various kinds of diseases that are involved in all the topics that I've shown above. Can I have the next slide? Yeah, so how do we do that? The ligand, when it binds, as you see on the top of the screen, changes the conformation of the receptor and enables one of the three kinds of G proteins to get activated, which downstream signaling leads to other proteins getting activated. So we, for example, can monitor ERK, as shown here, ERK phosphorylation, and see how is the efficiency of a peptide itself, natural peptide itself, a longer version of it or a shorter version of it, or a modified version of it, that eventually should help us create a drug candidate molecule. Can you go to the next slide? One click, please. One more. Okay, so here it is. The receptor is able to generate different kinds of signals so that the cell can respond differently. So the other molecule, which I mentioned one slide back, is what is called as the beta-arrestin receptor. So different diseases could be because of the differences in different downstream signaling molecules that are not functioning or working extra. Okay. In terms of the transmission of signal, you don't want signal even when there is no ligand. You don't want a signal that is in excess of what the ligand wants to convey, because the response will be predominantly based on how the signal is received. And if you can now generate a small molecule that will give a biased signal either to the left, the purple hexagonal molecules that are working in one downstream signaling, or the circular beta-arrestin, then you can control which disease to attack and how to attack. So G protein coupled receptors are the largest drug targets for all kinds of, about 30 to 40% or maybe 40 to 50% of the drugs that are available in the market target G protein coupled receptors. That shows how important they are. And now what I'm trying to tell you is that for this single receptor, we have the capacity to be able to read out what a molecule that we can generate, either absolutely novel peptide or a modified peptide from the naturally occurring molecule can generate, and how the cells will respond and what is the pathophysiological outcome of that. So we have a good readout for estimating the biological effect of various kinds of molecules. Can you go to the next slide, please? So here are the different ways of looking at it. And the extreme left is a radioactive labeled assay of the peptide. The peptide is radioactively labeled, and we can monitor how well the peptide is binding. And if you make a mutation in the peptide, does it change its efficiency of binding, which means its efficacy of action? How well will the peptide or the signal, even upon modification, elicit a response from the cell? So you can go on modifying and see that it binds well, but whether it will elicit a response as well or not, whether it will respond a different kind of response, is what we have the capacity to monitor. Now, the second, which is the colored slide, is showing a GFP labeled receptor, which means there is a fluorophore attached to the receptor. And what we are monitoring here is the internalization of the receptor. I told you in the beginning that the receptor is embedded in the membrane so that it can receive signals from outside. But if it is activated, chances are that it will actually go inside the cell, either recycle back to the membrane or just get degraded. So that is one assay of monitoring the efficacy of the ligand that we can do in our lab. The third from the left is what a ligand many a times elicits as a signal. The endoplasmic reticulum, which is another bag inside the cell, holds a lot of calcium, almost to toxic levels. But one of the ways in which signals are transmitted are that the downstream molecules elicit a release of calcium from this bag of calcium containing organelle. Okay? That calcium release binds to calcium binding proteins and switches them on, and you can have a response from the cell. So we now have the capacity to monitor how much calcium is released because of a ligand binding to the receptor, because of a new molecule binding to the receptor. The last to the figure to the rightmost is something that is quantitatively shown as a function of the ligand concentration as to how much calcium is released, which means how much response the cell is giving out depending on what kind of a molecule we are using. So we have tremendous capacity now to look at a library of molecules which are emanating from the peptide or otherwise that will show a response that could be connected back to which cell type we are using, which tissue type we are using, because it's expressed in many different kinds of organs, which means different kinds of tissues. And we can now do an assay that will show how good a drug candidate we have when we are able to generate one. And our goal, of course, is to generate a library of drug candidates that can be monitored for the response that cells give in disease conditions. Thank you so much. If you have any questions, I'll be happy to try and answer.
Thank you, Professor. I think we'll take questions right at the end. We'll quickly move on to the, thank you, Professor. We'll quickly move on to the next presentation. Professor Muralidharan, you are on.
Okay, good afternoon, everyone. First of all, let me thank the Dean is here and the team for giving this opportunity to present this work. Can you please move to the next slide, please? And also please have the contents because it was put under animation. So please bring all the contents together. Next one. Yes, that's all. Yeah, so as you can see, this particular slide is just presented here to give you a bird's eye view of different activities that we do. Our group is basically having strength in synthetic organic chemistry, and we use it to address the problems at the chemistry biology interface. And on the right hand side, upper side, you will see small molecules which are designed to target specific sites in proteins to have a beneficial therapeutic outcome They can be considered as ligands. And mainly by design, they have multi-targeting possibility, or they have, say, pharmaco for hybridization type of models. And mainly they are designed to target specific proteins to have a certain therapeutic effect. And this include also small molecules which can be used as chemical probes to understand different biological processes. And we also deal with the relatively linear oligomers, which have well-folded structure, and also segments which carry reactive groups like platinum-2 at specific locations. Such conformationally well-folded systems having a relatively higher molecular weight and size, they're actually designed to target macromolecular structures like DNA, to have a kind of DNA stapling effect, or to target protein-protein interfaces with well-folded systems like this. So all these, whatever you see on the right-hand side, are mainly small molecules designed and constructed through covalent synthesis. Covalent bonds maintain the structural integrity. And we also have projects which deals with molecules which self-assemble through non-covalent interactions to different functional systems. And I will discuss more about this kind of molecules here today because of their potential, translational potential in drug delivery. And they are glycolipid mimics, which have shown a lot of promising properties. So while this represent a discrete structure, as you know, there are supramolecular systems which involves assembly involving solvents, and there are a lot to understand because of the intricacies associated with the interactions and dynamics of interactions involving small molecules and the solvents. And we have actually designed the systems which respond through an on and off signaling dictated by hierarchical self-assembly of small molecules such as the one shown here. So this is a general outline of our activities. And I'm going to take you directly to two areas which we have come up with, and they are having translational potential. So I request to take to the next slide, please. So the first project I would like to present here is about chemical probes, which can be used for early detection of sepsis. And as you know, sepsis is a very, very serious medical condition, and it can be categorized as systemic inflammatory response syndrome, where the source is mainly a pathogen, a bacterial, fungal, or protozoal, etc. And what happens is that when the body responds to such an infection, and if that body's defense becomes dysregulated or uncontrolled, that defense will start causing damage to our own organs. And this goes through hyperinflammatory response or cytokine storm. And finally, our own defense will start damaging our own organs, and multiple organ failure leads to death. And this is a very, very serious condition. And especially those who are in ICUs or after organ transplants, for example. Can you please put the next image, please? Yeah. Those in ICUs after organ transplant, they are particularly vulnerable to sepsis, and this sepsis actually is a major cause of death in ICUs. And of course, early diagnosis is the key, because once you understand the progression of sepsis, we can start with appropriate medication early on and save the lives. So early detection of sepsis is a very, very important requirement. And since this is initiated through pathogens, there is a lot to worry, because we are actually living in an era where new infectious diseases appear, or new emerging or re-emerging infectious diseases are affecting everybody's life. And when such infections can actually start causing sepsis-like conditions, the mortality can be very, very high. Can you please go to the next? Please press the button one more time. Yeah. So WHO has recognized this as a global health priority. And in 2017, the estimates showed that about 48.9 million cases and 11 million deaths were directly related to sepsis. And actually, in terms of financial burden, more than $38 billion were spent on sepsis and related conditions. So all this makes sepsis a very, very high priority task. So let us see what we have done for the early detection of sepsis. Can you please go to the next slide? So here comes the importance of a few small molecules which we are all familiar with. And we know hydrogen sulfide as a rotten, smelling gas, and carbon monoxide as a poisoning gas, with poison effect, nitric oxide also. These are all gaseous small molecules which were known for wrong reasons. But in recent times, there is a lot of interest to understand the biochemical effects. And importantly, now we know that they actually mediate a lot of biological signaling processes, which are very, very important for the normal physiological well-being of every living organisms. And also their dynamic levels, like they are interdependent, and the dynamic variation in their levels is also extremely important in the pathophysiology of sepsis, because when after pathogen inversion, there's an overwhelmed defense and an excessive inflammation, cytokine storm, multi-organ dysfunction and deaths. And all these stages of sepsis, in fact, has a lot of direct influence by these gaseous small molecules. And which means that if we have a kind of method to understand the dynamic variation of these small analytes, bioanalytes, we will be able to see the progression of this condition and see whether the patient is actually prone to such a sepsis and septic shock. And actually, our work in these general areas have generated many IPRIs, and mainly these two are related to this particular process we are dealing with. Can you please go to the next image? So coming here, this is just a representation, is one of the props we have in hand. As you know, these are small molecules which are very short-lived in the system and are highly diffusible. So it is very important that they need to be measured accurately in the presence of various other analytes in the biological fluid. And if a person is in the ICU or post-operative ward, there should be a method to continuously monitor the variation in cancer transmitters. And this is the image which shows a very, very efficient probe we have designed and developed, which actually gives a multimodal response, because high accuracy is very, very important to make predictive models. And we could actually see use this molecule to have both Raman and fluorescence outputs. Can you please press the button 2 times? Yeah. And both these species can be actually used for real-time quantification of this analyte. So we have also some systems in our laboratory which actually can give a indication about oxidative and reductive responses in the biochemical system, and they can be monitored simultaneously. So our aim is to actually continuously monitor the redox status of biological fluids, such as blood plasma of a person in critical care units, so that an early prediction of sepsis can be made. And in fact, this work is actually being done in collaboration with Dr. Paul Ramesh in Apollo Hospitals and Dr. Ashish Kumar Singh in Mechanical Engineering Department, where he has developed some microfluidic devices also. So we deal with the chemical probes, in fact. And this is one area we hope that there's a very, very high translational potential. Can you please go to the next slide? And the second project I would like to present here is about cardiomyopathy and side effects during doxorubicin chemotherapy. So we are developing a system to modulate the pharmacodynamics of this particular drug to address these side effects. As we know, this is a very, very potent anti-cancer agent and used to treat various types of cancers, including solid tumors. One of the major problem is this cardiotoxicity. Please bring the next 2 contents also in the screen. Thank you. Yeah. So as I told you, cardiotoxicity is a very, very limiting factor here, and we cannot actually go for higher doses because of this particular effect, because the toxicity is directly related to the dose. And another thing is that this toxic side effects can manifest within days of treatment or even after 10 to 15 years of administration. That means those childhood survivors, there is a likely chance of getting cardiac problems at a later stage in their life. So it is extremely important to address this one. And this is just a pictorial representation of what happens, because this doxorubicin has a mechanism of action which involves intercalation into DNA, binding to the minor groove, and also stabilizing topoisomerase to DNA cleavage complex. And apart from that, this actually undergoes redox cycling, which generates large number of ROS, reactive oxygen species, and that is actually believed to be responsible for the oxidative stress and cardiomyocytes death. So methods to actually, control this pharmacodynamics will be very useful. So can you please press one more button? Yeah, next one. Yeah. So there are actually liposomal formulations like Doxil, which have shown very much improved therapeutic index because of this enhanced permeation retention effect in cancer tissues. There is, we achieved, it is actually available as formulation already in the market. So researchers are able to find better selectivity and reduced cardiotoxicity. So, but there is still scope for a lot of improvement if you look at the progression-free survival and overall survival of the patients. So can you please go to the next slide? So here comes our new drug delivery systems. Initially, we actually looked at a new class of lipids which contain a furanose-like head group. And the first one which is shown here is a natural lipid present in extremophilic membranes. And as a mimic of that, we have designed a large number of lipids. Some of them are shown here, which includes boloamphiphils and monopolar lipids. The interesting aspect is that we can actually modify the head groups or the tail with a very, very high selectivity. And one more image, please. They can be aggregated into solid lipid particles or vesicular structures. And if you put a fluorescent tag on the head group, you can actually see the ready permeation to the cancer cells in the form of solid lipid particles, or we can also use vesicular structures with the modification of the membrane periphery. So another interesting thing about these lipids is that they are very, very safe. And we have done flow cytometry studies to understand the cellular effect. And till 1 millimolar concentration, we found them to be very, very safe. And the IC50 values are much above 200 micromolar, well above 200 micromolar when we did the viability studies. So using these lipids, we have actually made we have loaded doxorubicin in a special group of lipids. The structures are not shown here, but we could actually achieve very good in terms of ROS generation in cardiomyocytes and cancer cells like A549 cells. We could actually see much difference the way ROS is generated. So we hope that this kind of carriers will be very much useful in addressing the cardiomyopathy, especially to improve the overall survival rate of the patients if it is developed further. And thank you very much for your kind attention.
Thank you, Professor Murali.
Yes.
We now move on to the next presentation. Professor Basavaraj, over to you. We're running slightly behind schedule, so request us to adhere to the time as much as possible. Thank you so much.
I'll try to do that. Yeah. Next slide, please. Yeah, the next one. So I broadly work in this area called soft and biological matter, wherein we typically deal with things like polymers, surfactants, and colloids, which are, I think, an integral part of any delivery system that people are trying to design in these years. Next slide. I think, yeah. So I think some of our contributions, can easily be into some sort of, oral liquid formulation. So we work with making formulations, microemulsion formulations and liposome formulations, which are very good ingredients for for encapsulating different hydrophilic and hydrophobic moieties, into the oral formulations. And as you see in the top, the plot here, the diameter of the droplet is a function of time. You can basically put in a small amount of water and have a water-soluble drug in these formulations, and then have them stable for an extended period of time. The liposomal formulations that Murli was also kind of mentioning, a while ago, these are again nano-sized compartments, which you can actually use it as delivery agents for both hydrophilic and hydrophobic moieties. And a lot of work that we have done in the recent years, if you look at the slide on the right hand, the image on the right hand side, we work with particular formulations called solid stabilized emulsions, which are basically emulsions or liquid and liquid dispersions stabilized by particles. And we can actually use, these are kind of systems where I can actually replace toxic surfactants and then use edible particles like polysaccharides, chitosan, and protein molecules, and stabilize liquid and liquid dispersions or emulsions. And again, you can basically have achieve extended stability, pH responsiveness, and thermal responsiveness, and so on and so forth. Okay, so therefore I think we have really good expertise in oral liquid formulations and helping people to develop new formulations and look at their stability and come up with ways to encapsulate new kind of molecules that people have developed for oral formulations. Next slide, please. So I just want to briefly mention about some nice devices that we have developed. This is work that has been done in collaboration with Professor Raghu Rangasamy. So this is a technique called spinning disk atomization, wherein you can actually use this technique for encapsulating both hydrophilic and hydrophobic drug, and convert them into a drug-loaded particle. So what you're looking at is a movie where there's a liquid that is dropping on the top of a rotating disc, and as the liquid basically rotates, you get these a liquid that basically breaks into smaller droplets. And all that you can do is you can basically collect these droplets into a container, and then you can think about some interesting chemical engineering principles like precipitation and other techniques to convert these liquid droplets into a solid loaded particles. And we have kind of developed techniques where we can look at metformin and itraconazole and both water soluble and oil soluble drugs to convert them into a particulate form. Next slide, please. So these are some microscopy images of the drug-loaded particles that we have developed. So this is a low magnification images to say that we can basically produce these particles in a very tight distribution, of the order of about 80 micrometer in this case. Of course, we can tune the droplet sizes as well as the final particle sizes. We can achieve drug-loaded particle size varying from about 10 micrometer all the way up to hundreds of micrometers. On the right-hand side, you can, this is basically a drug-loaded particle which has itraconazole plus an cellulose-based excipient. And, we are trying to currently work towards tuning the drug release rate from such drug-loaded particles. So maybe with that, I'll stop here. I'll be happy to take any questions.
Thank you very much, Professor Basavaraj, and to the entire team of panel 2. So due to lack of time, we will take all of the questions. There are some questions that have been posted, but we will respond to them over email. We will now quickly move on to panel 3, which is on healthcare equipments and infra.
Thank you.
Thank you. So over to Professor Mohan Shankar to start the session. Professor Mohan Shankar. Can you hear us, Professor?
Yeah, Padma, can you hear me?
Yes, yes, I can hear you now. Yes. Over to you, Professor. Please go ahead.
Yeah, Padma, can you hear me? No, I don't know whether I'm audible. So
yes, I can hear you. We can hear you.
Okay.
Yes,
Excellent.
yes. Please go ahead.
1 second. Screen is blocking this slide. I don't know which I'm seeing. Okay, so now here we have it. So welcome. I'm a faculty in electrical engineering and I head the Healthcare Technology Innovation Center. So since we are way over time, I'll try to wrap this in about 5 minutes. So can we have the next slide? Sorry. So simply put, I mean, IIT Madras is the leading medtech center in the country. These are numbers that sort of stand evidence to it. Even by global standards, this is truly a large ecosystem. HTIZ has done close to 300, 400 crores worth of projects, especially in the last 1 year in COVID. So we have commercialized 6 products that are currently global products. There are close to 300 publications, around 45 startups have been incubated, about 50 patents have been granted or pending. And we work with about 50 national, international collaborators, hospitals, industries, all kinds of industries, the Gs and Strikers of the world, all the way to startups that are booming, which you may not have heard of. Put all together, we have around 250 people. There are about 100, 120 engineers and doctors, about, as I said, 40 startups, 20, 25 industries, and we work across hospitals in the country. So next slide, please. And the point I wanted to make earlier is perhaps the only ecosystem in the country which has R&D, technology commercialization, entrepreneurship. Essentially, we take things all the way. And the dashboard, which I personally measure on what, are we really doing work that's worthwhile, is we count the number of people who are benefited by what we're doing. And it's very relevant to sort of the gathering here. So very happy to say the products that we have developed in partnership with industry and deployed in partnership with government agencies have benefited close to 5.5 to 6 million people. And that's something that we as IIT Madras and as a country, we're very proud of. There are various kinds of devices. I won't go into the details except to sort of run them through. There is this mobile surgical unit you might have heard of, done close to 20,000 surgeries till date. It's the only technology that has been approved by Government of India to operate outside a hospital. I'm very happy to say that it's actually operating for the last 10 years with 0 infections. And it's operated in really remote places. 2 ophthalmic devices, eye examination devices, both for adults as well as newborn. In fact, the newborn product has become a global standard product that WHO officially recommends it. Yeah, put it on presentation mode. Okay, fine. So the neonatal product, which you see here, the technology has many components to it, a very carefully designed optics so that it can actually work with a newborn. These newborns are as young as even 2 to 3 days, 2 to 3 weeks sometimes maximum. And this is a product that was completely developed from scratch in collaboration with the company and Narayana Nethralaya. And today it is a global standard product, meaning WHO refers to this product as the best product that low resource settings can use. So much so that it has actually displaced the products that are sort of developed by traditional legacy companies. So something that we can be very proud of. And so many devices like this have sort of come out. Not only these type of devices, very high value devices. And industry friends here will appreciate this because eventually you have to move to high value products in India. That's when your margins and your larger commercial play gets much stronger. So we have 2 products that are just about to enter the market. One is with Mitra Medical. It's the country's first indigenous endoscopy. You can imagine it's a very complex product, about 450 components. Everything you can think of in terms of complexity, biocompatibility, clinical safety, everything has been done through collaborators. This product is entering the market. In fact, a bronchoscopy product has already entered the market just as we are speaking. So I have not included that here. Then recently we completed an image-guided spine surgery system. in collaboration with the company. It's currently in cadaver trials. But for COVID, it probably would have entered clinical studies. So just to give you a sense of cross-section of the work which we have been doing. Next slide. So now, as you know, these type of devices, when you want to get it into the public health system, you literally have to do a project within a project. And people who have worked with public health system will understand this. So here is an example which we are currently doing. It's supported by Cognizant Foundation. We are covering 8 districts in Tamil Nadu, involving around a little more than 1,700 village health nurses, close to around 1,800 village health nurses. And this is done literally across the healthcare layer, starting from the health secretary all the way to the village health nurse. And this type of ability we have is what makes us very, very, very different from other institutions and other organizations. We can do this across the healthcare system and measure outcome in a formal method of healthcare technology assessment and so on. So this is just to give you an example of how deeply we are plugged into the public health system. And you will hear more about this in the next about 6 to 9 months in the public. Next slide, please. So again, not to give too many examples, this is the last example. So here is a truly a world-class biomedical research that is started in IIT Madras, which involves everything you can imagine: biology, bioengineering, instrumentation, clinical studies, animal studies. And this device today is actually entered the European clinical space. It was actually done, it has finished clinical studies in Europe, in Netherlands, and is on the way to get an FDA and CE mark. In fact, the 510(k) is being filed as we are speaking. So it just demonstrates what can possibly happen if you get really qualified people, and given the ecosystem IIT Madras has, we are able to convert scientific R&D to a global product. Next slide. So again, there's no time to go into the details except to say each of them is a story by itself. A couple of these companies have already gone global. I mean, in fact, 3 of them actually have global revenues coming out. So anytime you're interested in details, just write to me or just write to Padma. Somebody will connect you to us. Next slide. So I'll end there. Why are we successful? Why have we been able to make impact, which is probably the best in the country? Is we have an explicit focus on the healthcare impact and social impact. So yes, we do research, we do it successfully, but we are not satisfied with that. We really are looking at what impact it will cause in healthcare and socioeconomic impact. And we have extremely deep collaboration, both qualitatively and quantitatively. It's the best of the entire IIT system. We work with industries. We worked with almost close to 50 companies in the last 10 years. And we have an end-to-end approach. As you know, this healthcare play, you cannot say, I will do one piece of it and I want somebody to pick it up. The system is not that mature. So you really have to take an end-to-end approach, and we are very good at that. And the reason why we are good at all this is we have about 200 people who are really passionate about doing all this and getting it done. So thank you very much. I'll stop there and I'll have the rest of the panelists give their talks. Padma, over to you.
Yes, thank you so much, Professor. Over to now Professor Kavita Arunachalam from Engineering Design.
Good afternoon, everyone. I'm Kavita Arunachalam from Engineering Design Department. I'm an electrical engineer. In our lab, we work on microwaves and its applications. In today's talk, I'm going to give a summary of what we are working on using microwaves for cancer treatment. So you all know microwaves are used in daily life, but they are non-ionizing electromagnetic radiations with wide range of day-to-day applications in gadgets. What we are focusing is to harness this microwave energy, but in a more fine-tuned manner by developing devices that is capable of delivering focused power at the target, which is a cancerous tissue. So it's not just delivering the device. We have also perfected the means to maintain the temperature rise caused due to this high power focused electromagnetic wave. And we are looking at a narrow range for hypothermia treatment, which is 39 to 44 degrees that has to be maintained at steady state. The reason being that, next slide, please. So when you look at this picture, it is self-explanatory. There are several effects that happens when the treatment is done for a hypothermia. The cancer cells respond in different means, and as a result, it has been shown to be a very good radiosensitizer as well as a chemosensitizer. The good part is it does not have any acute or late effects. Clinical trials listed here shows that it excellently works for solid tumors. And coming to our focus, what we are working on is device development for breast cancer and cervical cancer. These are the most common cancer among women, and most of these cancers are detected at late stage, large solid bulk tumors, which has poor treatment outcomes, and there are no effective adjuvant therapies. So the question here is, why not use this hypothermia therapy treatment, which is used elsewhere in the Western countries? So the reason is, commercially, it is not available, whatever devices are in the market, and technology to target and deliver this heat in a better way for large solid tumors is lacking currently, which is what is needed for our country. Next slide, please. So this is one of the prototype we have developed. This is a work under development. The second prototype we have come up with for treating locally advanced breast cancer. So this technology makes use of radar technology, where you use an array of antennas to steer the beam anywhere inside the breast. To do that, we have not only developed miniaturized antennas, but also a complete treatment planning approach, starting from the image till the treatment. We have developed the in-house algorithm, which is also validated in a wide range of patient models. We also developed a rugged source to deliver this targeted heating. So now we are at a stage where we have working on clinical translation of this device, because the algorithm and the source are available. We want to work on the patient treatment table. So this is in the clinical translation right now. Next slide, please. So the other one that we have developed is also on the cancer, which is for cervical cancer, locally advanced cervical cancer. Typically, for complete better response, they conclude the treatment with high dose rate brachytherapy. So here we want to add hypothermia along with brachytherapy. So instead of doing the procedure twice, we have developed applicator, which is 2-in-1, where we can deliver both. So radiologically, we have assessed it. Experimentally, we have shown. Now we are taking it's undergoing quality assurance testing. This work is done in collaboration with CMC. Next slide, please. So the related focus area is, having developed these devices for hypothermia treatment delivery, we are now moving towards image guidance, so that we have less dependence on the CT image, which is again radiation exposure will increase. So this work is done in collaboration with applied mechanics faculty Arun Tittai. So we are working together on integrating ultrasound with our device, so that we can develop a low-cost standalone imaging treatment delivery setup. So this is what we are working. So twofold. One is it will help in us treatment planning, and also provide thermometry. So we're using ultrasound for thermometry as well. Lastly, we see that nowadays the immunotherapy and molecules for targeted therapy are coming, because it's better than a conventional systemic chemotherapy. But the study shows that not all tumors are responding to these molecules, and people are working on developing molecules which will enable a cold tumor to become hot. So since hypothermia causes cell death and also modulates the tumor microenvironment, which helps release the antigens in the cancer cells, we are working on preclinical investigations to study if it can be assessed in preclinical models in small animals. This work we're hoping to start with in collaboration with biotechnology department at IIT Madras, Professor Raila. And the another outcome that we see is in the process, we have to do a lot of miniaturization because we have to customize and develop a protocol for small animals. And the same technology can be used not only for understanding how it works in the preclinical model, but also to develop device for vet medicine. So currently for veterinary medicine, if an animal is succumbing to cancer, there are no options. So this is what we are working on. I'll take questions later on. Thank you very much.
Thank you very much, Professor Kavita. Now we're moving on to Professor Nirav Patel.
Yes, hi. Good evening, everybody. I'm Nirav Patel. I head Inspire Lab at IIT Madras in the Department of Engineering Design. So I'll present some of the works that we do in medical robotics. So I have extensively worked in the domain of medical robotics and particularly image-guided surgery for the last 10 years during my postdoctoral and PhD experience. And my focus here is actually to develop low-cost image-guided robotic systems that use one or the other imaging modality, ultrasound, CT, or in future MRI. And then the focus is on particular interventions. So these are minimally invasive procedures such as biopsy or ablation. And so we do development at all levels, from the prototype to all the way taking the devices to the operating room. Next click, please. The focus of the lab is actually on clinical translation. So whatever devices we make, we try to get them out of the lab. That's one of the things that I always tell students as well, is optimize them for the clinical application that we're implementing these devices or developing them. And so the overall focus is clinical translation. Next slide, please. One of the systems that we are working on is for CT-guided brain biopsy. While the present system is being developed for brain biopsy, it could be used for other applications as well. When we look at brain tumors, the number that we find over here is 24,000 people die everywhere in India, but that's an underreported number because we don't really have a very strong cancer registry in all the states. To diagnose these, typically a brain biopsy is taken using a bulky mechanical frame, and that costs 75 lakh rupees, just the frame. And it makes it less accessible to a large section of the society. Furthermore, when this has to be treated, if it is cancerous, it basically requires removing the tumor through craniotomy, which is very invasive. And sometimes during the procedure, because you have to have margins, they have to end up removing, in fact, healthy brain tissue. So the post-surgery quality of life is quite bad for many patients. So taking this into account, we have been working on development of a robot that can be used inside the CT scanner. So we could actually target the tumor more precisely. It can be taken further for MR guidance in the future as well, but that is something that I think we as a country are not ready yet. CT guidance is becoming more popular. Hopefully in next 5 years, we will be looking at MR guidance as well. But one of the outcomes that we look over here is that the bulky mechanical frame results in longer procedure time. So if we make procedure time shorter, we can squeeze in more patients. And so that is one way to look at that. How do we convince hospitals to go for a more tax-heavy solution that might sound complex, but it would be economical in a long term. And this will make the cure more accessible, basically. And so this has a lot of potential for minimally invasive treatment as well if we go for either ultrasound or laser ablations of tumors. So this is one project where we have got funding from BIRAC and it's in early phases of development, but in a year we'll have a prototype that will have been validated in basically a clinically relevant setup on phantoms and baby animals, but that's the current state of this project. Next slide, please.
Professor, you have 2 minutes.
Sure. So the second system that we're developing is for ultrasound guided interventions, where we want to use ultrasound being a portable and low cost imaging modality. So we take this system where we are building a platform basically that could allow us to precisely place either diagnostic or therapeutic instruments such as ablation probes or needles precisely into any abdominal region or organ basically. That's the overall idea. Next slide, please. The third project that we have just initiated kind of is on early detection of breast cancer. Breast cancer is one of the, I would say, major concerns when it comes to cancer in India. 1 out of 8 women suffers from cancer globally, and the mortality rates are pretty bad too. One of the important things is actually early detection. So if breast cancer can be detected early, it could actually increase the survival rate by a big margin. So if you can see here, 5-year survival rate decreases by 2.7 times if detected at stage 4 instead of stage 1. Unfortunately, because of the social taboo and lack of awareness, the breast cancer screening is quite poor in India. Only 30% of the reported cases that we get are diagnosed in early stage. And so our goal is to develop a screening system. Next, can you please press the key? Okay, so the idea came from this very simple system that I have seen across many places is blood pressure monitoring, right? This is a kiosk. This is doesn't require any human interaction, basically. It becomes more complex in case of breast cancer detection because it has to have palpation or use a sound-based scanning system that allow the woman, in particularly the rural areas, to break the social taboo and be more aware that there is a device that doesn't require any human interaction, basically, and go themselves get screened. Next slide, please. This is the overall aim for this project, is to develop a kiosk where women can really walk in there. There will be a very simple interface where they stand there, and the system eventually, the beginning it would of course would require some assistance from the person to be scanned, but eventually it would be an autonomous system where it would give a graded score from 1 to 10 and say that, okay, do you need further screening or not? If it gives a certain score, they would be requested to go and get them screened either through mammogram or through palpation by a trained physician. And so we are just formalizing now this idea into early prototype, basically, where you want to see what are the possible ways to minimize the human intervention. You know, the healthcare professional, if they don't have to interact with these women in the rural areas, it might make it much more accessible. And so this is something that we are going to initiate very soon. Next slide, please. Yeah, thank you all. Thank you very much. And that's all from me. Thank you.
Thank you, Professor Nirav. Over to Professor Bijo, and request Professor to, kind of keep it as brief as possible. Thank you so much.
Thank you, Padma. I hope you can hear me.
Yes, we can hear you, Professor.
All right. Hello, everyone. Good afternoon. My name is Bijo Sebastian. I'm a newly joined assistant professor in the Department of Engineering Design. I'm primarily a mechanical engineer, mostly focusing on design, analysis, integration, and testing of mechatronic and field robotic systems. Most of this research involves concepts from multi-body dynamics, modern control theory, and robot motion planning. Some of the specific applications of these techniques is towards design and integration of exoskeleton gloves for rehabilitation, autonomous navigation mostly in indoor and outdoor environments, multi-robot handling, multi-robot material handling systems, and improvement of ADAS systems. So among most of my research areas, exoskeletons is one topic that I work towards the healthcare side, and we would mostly look at that through the rest of my slides. So exoskeletons are usually devices that are used to assist human beings, either by improving. Can you pause the video for a second? Thank you. Yeah, so exoskeletons are essentially devices used to help assist human beings. Over the last few years, some of these devices are being used mostly in medical rehabilitation, mostly as a replacement for physiotherapy. So one use case would be in the case of patients who have suffered either injuries to their brachial plexus nerve center or after stroke. In such patients, sometimes they lose control of their hand muscles. Existing medical studies state that over a long duration of physiotherapy, they can regain back control of their hand. So as I mentioned, this involves time-consuming and costly physiotherapy because physiotherapy often requires a trained physiotherapist to be with the patient throughout the duration of training. The goal of using exoskeletons is primarily to avoid the requirement of a trained physiotherapist. Basically, exoskeleton gloves are devices that the patient can put on their hand, and then this device can assist the patient in performing therapeutic exercises. Please go ahead and play the video. This video shows one similar exoskeleton system. This was developed at the Robotics and Mechatronics Lab at Virginia Tech. The video, when it plays, will show the system mounted on a wooden mannequin. It's not a real human hand. And enforcing something called as a cylindrical grasp. Next slide, please. That's another example. We can go on to the next slide. All right. So one of the disadvantages or limitations of existing exoskeleton systems is that, like the one that we just saw in the video, is they are often bulky and slightly scary looking designs, which patients are not always comfortable using. So one of the approaches that I have explored through some of my research is how to streamline some of these systems so that they are more approachable, less bulky, and as well as more user-friendly. So one such design in its initial ideation form is shown in this video. Please click on the picture to play a video. Yep. The equations on the side basically describe how the optimization was done to develop this mechanism. But as the video shows, this is a double 4-bar mechanism which mimics the motion of a human finger, basically the flexion and extension. Go to the next slide. Once more. Next slide, please. Yeah. So this was the very first version of the prototype that we built. This was go back once. Thank you. Yeah, you can click on the picture on the right side. That will play a video. Yeah. So the video shows the working of the initial design. It came with a computer graphical user interface. Basically, by providing instructions from the computer, the pneumatically powered glove will enforce a pincer grasp. Let's go to the next slide. And this was the same single degree of freedom mechanism replicated for all 5 fingers. Also, we replaced the pneumatic actuation with electric actuation because pneumatic actuation means you have to carry around a pneumatic cylinder with pressurized air, whereas electric actuation system only requires a lithium polymer battery and it reduces the weight, it makes the device more portable. Next slide, please. And this is the state-of-the-art system that we currently have. Single degree of freedom mechanism replicated on all 5 fingers, allowing you to do different kinds of grasp as shown on this slide, shown on the right side. Next slide, please. All right, so everything that I talked about till now was work done as part of my doctoral research at Virginia Tech. After joining here at IIT Madras last November, ongoing research within the engineering design department focuses on some of the aspects listed here. First and foremost, we are trying to modify the existing single degree of freedom design to 2 degrees of freedom design, which means instead of just being able to do a cylindrical grasp, the glove will be able to do additional kinds of grasp, such as the kind of grasp you use when you pick up a phone or pick up a textbook. Another approach is, rather than using a computer or an embedded device to trigger open and close of fingers, we are trying to go for a more intelligent control. Basically, the user should be able to trigger the device. Depending upon how the user tries to move his or her hand, the glove should understand what's the type of grasp and guide the hand towards it. Again, we are trying to go for a cheap, portable, lightweight system with at least full day of battery backup for the patient population here. The devices that I just showed in the previous videos were all tested on either mine or some of my colleagues' hands. So it's always tested on healthy human hands. What we are trying to go for is clinical testing with medical partnership, with the overarching goal being to deliver a viable product for the Indian patient population. That's all I had to present. I'm happy to answer any questions that you might have.
Thank you very much, Professor Bijoy, and to the entire panel 3 team. In the interest of time, we will wrap up with the panels at this point in time. And it's, I think, overall been a fantastic program and very engaging questions, very promising projects. I would now like to warmly invite Professor Mahesh Panchagnula, who's our Dean, Alumni and Corporate Relations, to share the closing remarks for the day. Over to you, Professor.
Thank you, Padma. Thank you. Good afternoon to all the panelists and the attendees. This has been, I've been on the sidelines watching some of the talks, and it's wonderful to see the set of projects in a very essential space, healthcare, originating from IIT Madras. I'm sure our partners from the companies who are attending this afternoon's webinars would have also picked up some cues, picked up some interesting ideas for them to think about and come back to us with possible engagement opportunities. Healthcare is in a space where it is today indispensable to not use technology. There is no scope. We have to rely very heavily on technology, in order to realize bedside outcomes that are tangibly better than what we were able to do yesterday. So in that sense, an institution like IIT Madras, with the breadth of technology available here, is almost an essential partner in this growth process, where we try to create healthcare outcomes that are tangibly different day after day, week after week. And investing CSR dollars to CSR rupees to help that process along, to nudge that process along, is very important. While our society heavily relies on profitability as the driving force to enable a better life for all of us, there are spaces in healthcare, particularly, where an initial nudge from a not-for-profit source of money is essential. That nudge can come in one of many ways. That nudge can come in the form of taking a lab scale prototype and helping us scale it out into a pilot situation where we demonstrate the profitability aspect of that innovation or intervention. That nudge can come in the form of even helping develop this new kind of a prototype, which is squarely in the realm of government funding today. We believe that the use of CSR money to help create a better technology for healthcare providers tomorrow is essential to a better tomorrow. And relying squarely on the government for that initial few TRL levels of development is also not the best possible solution. CSR money invested in that space where you are creating prototypical solutions and helping create either new prototypical solutions or taking existing prototypical solutions and helping deploy a small pilot are absolutely viable, perfect, and legitimate uses of CSR rupees. So I would encourage our attendees to think about this angle of investing their CSR. As we have sort of learned now, it is important that we, let's say, distribute a whole bunch of ultrasonic imaging machines for, let's say, a certain set of rural areas to benefit from. But it is equally important in a country like India to investing in the development of, say, a low-cost ultrasound device that can tomorrow better the lives of a hundredfold increase in the number of patients that could be impacted by the same amount of invested rupees in that intervention. So creating a better future for all of us through investing CSR in technology development is absolutely essential for a country like India, which is now slowly going beyond the realm of, let's say, needing to feed the hungry to creating technologies that would be more efficient at delivering food or creating a more nutritious supplements. So with that, I'll close my remarks. We have a whole range of technologies that, as you would have seen, our faculty are pursuing, from the Department of Engineering Design, Department of Applied Mechanics, Department of Electrical Engineering, our Healthcare Technology Innovation Center, as well as several others, including humanities, where healthcare policy is an active area of research. So please come on board. You will find whatever be your flavor of where you want your CSR money spent, you would find it quite actively pursued here at IIT Madras and to your satisfaction. So we look forward to engaging with as many of our corporate partners as would be interested in this space. Thank you, Padma, and over to you.
Thank you very much, Professor Mahesh, for those well-rounded remarks. And thank you one and all for joining us this afternoon. Thank you to all of the professors who have made your time out to be part of this program today. We hope and we believe that it has been very fruitful and meaningful to each one of you. We look forward to continuing this engagement. Thank you very much. Bye and have a good evening. Bye-bye.